the Sanger instrument, I can get up to 800 base pair fragments read, and given that the size of my genome is 3 billion base pairs, I would need, what? 100 years?
The Human Genome Project generated the first reference sequence of the human genome, representing approximately 3.1 to 3.2 billion base pairs. The project began in 1990, produced a draft genome in 2001, and by 2003 had generated a high-quality reference covering approximately 92% of the genome. What remained were highly repetitive regions that were difficult to sequence. In 2020, the first gap-free human genome assembly was completed, marking the culmination of more than 3 decades of work. Today, thanks to next-generation sequencing, or NGS, an entire human genome can be sequenced in about a day, although the complete lab workflow, which includes things like sample preparation, sequencing, and data analysis, typically takes longer. This dramatic improvement in speed is possible because NGS sequences millions to billions of DNA fragments at the same time. In contrast, Sanger sequencing analyzes individual DNA fragments in separate reactions, making it far less suitable for large-scale genome sequencing. Many NGS workflows align sequencing reads to an existing reference genome, such as the human reference genome produced through the Human Genome Project. However, NGS can also be used for de novo genome assembly when no reference genome exists. The basic principle behind NGS is that DNA is fragmented into many short pieces, each of which is sequenced independently. The resulting sequences, known as reads, are then computationally assembled or aligned to reconstruct the original DNA sequence. NGS can be used to sequence both DNA and RNA. The workflow begins with sample collection, followed by purification of DNA or RNA. The nucleic acids are then assessed to ensure they are of sufficient quality and are not degraded. For most RNA sequencing applications, RNA is first converted into complementary DNA, or cDNA. This is just because the instrument and the polymerases that will be used can only handle DNA. The cDNA preparation is achieved through an enzyme called reverse transcriptase. Then what is called a sequencing library is prepared. This is where the DNA or cDNA is fragmented into many shorter pieces from the longer DNA molecule. To put this in context, if you extracted the entire DNA from a cell and you stretched it, it would be about 1.82 meters. That's quite long, and most instruments cannot handle that length of DNA. So what happens is it gets fragmented into smaller pieces that are then sequenced. You generate the fragments using mechanical means like high-frequency sound waves, or you can generate the fragments by enzymatic fragmentation. Next, specialized DNA sequences called adapters are then ligated to both ends of each DNA fragment. These adapters contain the sequences required for binding to a surface that will be used for sequencing, typically called a flow cell if you're using something like the Illumina platform. Next, you have the sequencing primers that are able to bind to these sites, and then the sample indices. If you have more than one sample, say you've got a control and a treatment, but you need to sequence them together, there are ways to do it, and this is when you introduce something called an index. The index will go onto each of the samples that belong to one group, each of the library, the fragments, so that when you go to analyze, you know which ones came from which. In some workflows, you may also have to introduce unique molecular identifiers. So after your ligation, excess unbound adapters are removed, and now you have a complete library. But depending on the application, the library may have to undergo PCR amplification to increase the amount of DNA available for sequencing. So think of it as if you want to detect a signal. If there's just one person shouting, it's harder to hear than if you have 100 people all shouting at the same time. So this is when the library will just be amplified so that whatever's happening, it can be detected instead of just having one molecule or 2 or 3 or 4 molecules. Obviously, PCR can introduce some biases, so actually modern sequencers have worked towards removing this part where you have to amplify via PCR before you can detect. There's some quality control that has to happen. So before sequencing, the library is assessed to confirm that the fragment size distribution is suitable for the platform that you're using. So something like Sanger, you would have a limitation on the size that is ideal, or Illumina platforms or other platforms may require different fragments. And the longer read platforms would want really high quality RNA that are quite long and not fragmented as much. You also want to check the concentration, because if you don't have much of it, then during the sequencing things will go wrong. One of the most widely used NGS technologies is Illumina sequencing. This one uses a method known as sequencing by synthesis. The hero of all of this, you must remember, is DNA polymerase. So DNA polymerase is going to go along and copy your DNA. And as it copies it, you are able to determine what the sequences were, because if there's an adenine, it will add a thymine. If there's a guanine, it will add a cytosine, and that's how you're able to determine what's in your sample. Let's leave it here for part 1 and continue to work out how the rest of the sequencing occurs in the subsequent parts. If you're doing sequencing, you likely would want to look at other topics such as primers, and so I've got a tutorial on here about primers. I mentioned quality control, so you can have a look on here for a tutorial on a bioanalyzer, how to check the fragment sizes as well as the quantities if you want to use a nanodrop or a bioanalyzer. Thank you so much for checking out the video. I really appreciate you subscribing to our study group and giving the video a thumbs up. And I'm looking forward to catching you in the next video. God bless. Bye.